Meiotic drive is a phenomenon in which an allele is transmitted to more than the expected 50% of offspring, distorting normal Mendelian inheritance. Under Mendel's law of segregation, the two alleles of a gene have an equal probability of being transmitted to the gametes. Meiotic drive breaks this expectation by allowing one allele, known as a meiotic driver, to gain a transmission advantage over its alternative. Importantly, a driver does not necessarily benefit the organism carrying it. It may even be harmful to the host, yet still spread through a population because it increases its own transmission. Meiotic drive can occur through both asymmetric and symmetric mechanisms. In asymmetric drive, the meiotic products have unequal reproductive potential. This is particularly important during female meiosis, where only one of the four meiotic products become the functional egg, while others become polar bodies. In symmetric drive, the meiotic products have more equal reproductive potential, so a driver must use other mechanisms to gain a transmission advantage. One example of asymmetric drive occurs in mice, where certain centromeres can exploit the asymmetry of the meiotic spindle, increasing their probability of being transmitted into the egg rather than a polar body. A similar example occurs in maize, where heterochromatic chromosome knobs can exploit spindle asymmetry to favor their transmission. An example of symmetric drive occurs in Drosophila simulans, where an X-linked driver disrupts the segregation of the Y chromosome during the second meiotic division. This causes Y-bearing sperm to fail to develop properly, so males produce predominantly functional X-bearing sperm and therefore produce almost entirely female offspring. Although the total number of functional sperm is reduced, the X-linked driver gains a transmission advantage. Drive does not always act during meiosis itself. Post-meiotic drive occurs when a driver selectively affects gametes after they have been formed. A classic example is the mouse T haplotype, a selfish genetic element found in mus musculus. In males carrying one copy of the T haplotype and one normal chromosome, genes within the T haplotype impair the development and motility of sperm carrying the normal chromosome. As a result, sperm carrying the T haplotype remain more functional and have a greater chance of successfully reaching and fertilizing the egg, allowing T to be transmitted to well over 50% of offspring. However, this transmission advantage comes at a cost. Males carrying two copies of the T haplotype are generally sterile. Meiotic drive can therefore have important consequences for populations. A transmission advantage does not necessarily come without a cost. As seen with the T haplotype, drive can reduce fertility, while sex chromosome drive can distort the sex ratio of offspring. For example, a drive that preferentially increases the transmission of an X chromosome can produce strongly female-biased offspring. If such distortion becomes pronounced, it can affect the reproductive dynamics of the population. These fitness costs can create an evolutionary arms race between the driver and the rest of the genome. Natural selection may favor suppressor genes that reduce the driver's effects or restore more normal transmission. The driver may then evolve mechanisms to overcome suppression, while suppressors evolve in response. This cycle of driver and suppressor evolution can continue over many generations. The evolutionary arms race also highlights how meiotic drive differs from ordinary natural selection. Natural selection changes allele frequencies because differences in traits affect survival or reproductive success. Meiotic drive changes allele frequencies by distorting which alleles are transmitted during gamete formation or development. In other words, natural selection acts through differences in organismal fitness, whereas meiotic drive can act directly on the transmission of genetic material. Meiotic drive is also different from engineered gene drives, which are deliberately designed by humans to increase the inheritance of a chosen genetic element. For example, researchers have engineered gene drives in malaria mosquitoes to spread genetic changes that reduce mosquito populations or interfere with their ability to transmit malaria. Unlike naturally occurring meiotic drive, these gene drives are deliberately constructed to bias inheritance. Meiotic drive shows that Mendel's 50-50 expectation is not an absolute rule. While Mendelian segregation normally gives competing alleles an equal chance of transmission, selfish genetic elements can evolve a mechanism that bias their own transmission. In doing so, they reveal an important principle of evolution. The interests of a gene and the interests of the organism carrying it do not always have to be the same. Now, test your understanding by trying the questions on this topic. And if you've enjoyed the video, don't forget to like, subscribe, and share to support the channel. Thank you for watching.